Thank you much for the invitation, for the educational work package. The title of this mini symposium is also the limitations of precision oncology. So I want to talk about that a bit more. But first, I like to start with two little stories. We are here at Marie Slobodska's difficult to pronounce Curie Institute, and of course I googled if she had anything to do with Leiden, where I come from. And there was a little story I found that you probably all know that she died of aplastic anemia because of her work. And she invented polonium, named after Poland, and she worked with radium. And she wanted to do research in the cold climate, a very cold laboratory, and that was available in Leiden. So at that time she lived in Paris, and she took the train with the radium in her suitcase to Leiden. So probably other people died of her work as well. But so that's a little story about her. But she's a female with 2 Nobel Prizes, so that's really memorable, and it's nice to be here for the second time in my life. So yeah, so I have some disclosures, and the second one has to do with what you were talking about, because I'm on a committee that evaluates the societal worth of new interventions and new medication, and I've learned a lot over the last 2 years. Fair innings was a new word to me. You explained in a nice slide, but it also shows the other side of a medical oncologist wanting the best for his patients, but also the needs of a society with a growing healthcare problem. Where I think 1 out of 3 people should work for healthcare if we continue to go like that in a couple decades. So this is again educational session. So I start with personalized cancer medicine. Sir Beatson, and he was a surgeon in England, born in Australia, and he was the first one to do that, and you can go back to his Lancet paper, I think in 1892, you can still find it in the internet, or 96. And he as a farmer boy, he knew about if you took away the ovaries in cows, that it did something with the milk production. So he had patients with locally advanced breast cancer, and there was no treatment, and he took away the ovaries, and the breast cancer got better for a while. So he was the first person, a surgeon, to do personalized cancer medicine ever. And then later in the 60s, tamoxifen was developed as an anti-birth control pill. Didn't work very well, but it was then being used also for breast cancer. In the 1980s, they found out that there were hormone receptors, so that was really the first personalized treatment. Chemotherapy was, and we still is, the cornerstone of treatment of cancer patients with lymphomas, for instance, and testicular cancer. But in many other cancers, there was a ceiling that we reached around the end of the last century. So at that time we had the first treatments, imatinib, Herceptin, that were really effective, and there was a lot of hopes and hypes around that. And at the same time, over the last 2 decades, and we have heard about that today, there were significant advances in molecular diagnostics. There was an increasing molecular subtyping of cancer. Lung cancer was the first one. And there was an increasing availability of molecular targeted agents. So the setting was right for personalized cancer medicine. On the other hand, there were also people that were critics, and they said, okay, yeah, you did, you cured almost GIST and CML, and you doubled the survival of HER2-positive breast cancer, but that's it. And you cannot go further. The low-hanging fruit has been taken, and there were a flow of early personalized cancer medicine trials, the basket trials, like the vemurafenib trial, There was one basket for multiple cancer types. The umbrella trials, we had, for instance, one disease like lung cancer with all these different molecular alterations, and then the platform trials like the STAMPEDE study and the studies that we're talking about today. But then the first published personalized cancer medicine trial with a control arm was negative. It was a French trial, and there was randomization, molecular targeted agent versus treatment at physician's choice on the left hand. Primary endpoint was negative. But on the secondary endpoints, you can see that for subtypes, there was a positive outcome. So that means that you have to stratify. There was harsh criticism from key opinion leaders. The Shiva trial was the biggest, 741 patients. But as you can see, from the almost 500 patients, only 293 had a mutation, which is a bit lower than we see now. But only 96 received matching drugs. So that is a low percentage. And if you go down, like here, you see that 26, 10, 13%, 4%, 4%. So it's important that you have enough drugs if you do a personalized cancer medicine trial, because if you find the genetic alteration and you cannot treat the patient, the trial is bound to be negative. So that's important. And so as I said, there was nihilism. There was lots of people said, okay, this is not going to work. That's only 8 years ago. But there was also enthusiasm from diagnostic companies, pharmaceutical companies, patients, some physicians, believers, and there was progress also in lung cancer. So what was there to bridge in order to start a more realistic approach? It's important then to have a better understanding of molecular diagnostics in relation to outcome. So the new research needs to be done, databases need to be built. We should understand who is responding and who is not. Access to sufficient drug, like I said, and larger scale collaborations are needed. So a few slides about the Netherlands, how it started, and two initiatives came together. Maybe I have to start with the bottom one, with the Hartwig Institute. Was founded paid by an anonymous person that gave a lot of money. We were happy with that. So we could do a whole genome screening for patients in the Netherlands. It was done on a place not linked to a university, because we knew all the problems. It was known that the egos are big in the university world, so that was a separate place. And there was centralized whole genome screening, a centralized database, clinical data were kept, and a report was generated really speedy within 2 weeks, and then maybe after 5 days later on. So it's important to have speed. Then there was also the Center of Personalized Cancer Treatment, CPCT, which is still there. And that had the ambition to, it was an academic initiative to bring all the database together with the clinical data, and almost all hospitals in the Netherlands are now participating. So the first study that was done was just to take a biopsy before standard treatment in a cancer patient that had palliative treatment. And we collected the clinical data and the whole genome sequencing data. We found that a lot of patients had an actionable event, some outside and some inside the indication. And especially the patients outside the indications were the patients that were problematic, because unlike in the US and in Switzerland, I believe, we could not give expensive medication outside the registered indication. So what to do? So therefore the DRUP study was founded. It's a very lean organization. I saw the millions in Norway that built that trial. Here it was very lean. There was a organization, Barcode for Life, that bicycled up a mountain in France each year, bringing in 1 million, and there was the Dutch Cancer Society grant of about 1 million per year. And the companies paid a fee per patient, around 5,000. But still, it was a lean organization for refractory metastatic cancer patients with an approved target, and the primary endpoint was a 60-week progression-free survival, and the patient that entered had progressive disease. And of course, safety and access to data, etc., were secondary endpoints. So you have seen these kind of slides more often. I want to emphasize a couple things that was unique to this trial. Of course, you could enter the trial, 8 patients, is 1 or more patients had benefit, you do another 16 patients, so you end up with 24 patients. And when the cohort is closed, we do the whole genome sequencing and correlate that with outcome. If the trial was negative on 24 patients, and if you see a molecular profile that fits better to the responders, you can go back to stage 2 again. But if you have 5 or more patients being successful, then you can go to this risk-sharing model, where the pharmaceutical companies pitch in for the first 4 months. And of course, their benefit could be that this may lead to registration in the future. And the insurance companies, they pay for prolonged treatment after 4 months. So that's the pay for performance model. So the key to success is, how does one, and I would say a physician in any hospital, know what to test? That's important. Hopefully a more broader test. For that reason, we are making, in the past you had these ESMO minimal clinical requirements, maybe you remember those, and they were changed into the guidelines. And the guidelines may be out of date a bit. So we made per tumor type exactly what needs to be tested in order for a patient to get to regular medication. But of course, molecular testing is also done for diagnostic purposes and for prognostic purposes. So it's quite a difficult situation. We would like to go to the Norwegian model to do at least broad testing for the patients in need of treatment. But it's not easy. We're working on it. And then it's important to have MTBs and to educate the physicians and the public, and that's what we're doing now. So key to success is the availability of drugs, many drugs, as you can see. And the second stage agnostic cohort that was filled up was the MSI positive tumors with a clinical benefit rate of 63% and 40% objective response rate, and prolonged clinical benefit in some patients. So this cohort proceeded to the 3rd stage. And the uniqueness about this is that before the study had ended, we did a pre-planned interim analysis and gave the raw data to the healthcare authorities. And they did an independent review of our data, and they came out with an overall response rate of 44% and not reached median duration of response. So in our country, the medical oncologist at that time had a system in place to judge single-arm studies on their value, and there were 2 conditions that were important. The disease had to be rare, so 6 per 100,000 or less, and the mechanism of action of the drug should be understood, well, which was the case here. And as you can see, the overall response rate and the duration of response is well within the boundaries, and that led to the reimbursement, full reimbursement of nivolumab for MSI-positive tumors. So this is the model that we would like to use more, because if there's no model afterwards, there's no need doing these kind of trials. So the current status is we have more than 2,500 patients included and 1,500 started treatment. We have 33% clinical benefit rate, which is a bit lower than the Norwegian example that you just heard. And we are serving equally rare and non-rare cancers. And of course, the non-rare cancers have rare mutations or variations, so it's both rare. So the next stage 3 cohort is PARP inhibition, olaparib in BRCA-mutated tumors. The stage 2 was published 2 years ago already. The third stage now, because this is so rare outside the registered indication, that we would like to do this on a European level. The third stage has started in Norway and will probably start really soon in the next months in the Netherlands, and we hope to broaden this through the network. It's also important to publish negative cohorts, because we're talking about molecular diagnostics and novel treatments, but it's just as important to do this and to find out, to save money for the society, to find out that you should not treat a patient with a high mutational load in colorectal and gastroesophageal cancer. So that's really important, and that may differ per tumor type, because other tumor types did expand to stage 2. But we also found a signature, for instance, for MSI-positive tumors with nivolumab. That was as an abstract at ASCO. We found, for instance, KRAS G12C as a predictor for LONSERV activity. So you can learn much, and you can also save money by doing these kind of exercises. So the future of DRUP in the Netherlands and abroad is we also work on a combination targeted treatment if the patient has 2 variations or more. We also look at treating in earlier lines of therapy. We have this drug access protocol with early access to generate data. We do this for NTRK, for instance. And we have multi-stakeholder collaborations with the health care organizations and the payers. There is an existing collaboration with the US and Canada, but it's not as easy as the collaboration that we have in Europe. So it's always a lot of people, and it's important. It's a lean organization. We only pay €1,200 per patient, mainly for the biopsy. That's not standard of care. And for the CRFs. And we have a lot of PhD students doing actually a lot of work looking at the patient data, responding to requests from all the physicians. And we have, of course, we do our own CRO, and so it's really an academic exercise. And we're happy to have IDMC and a steering committee. So I just explained to you how we went from CPCT, Hartwig, DRUP, in collaboration with the payers to this risk sharing and full reimbursement. And you just have heard how it worked in Norway, and we are now trying to do this on a European level. So PCM4EU. 17 partners. It was mandatory to have at least 16 countries. We managed to do so. And it's funded by EU for Health, which is a project under Europe's Beating Cancer Plan. This is how it looks like. There's a website. You can find it all here, all the information. For package 1 is management and questionnaires that we will send around. 2 is about molecular diagnostics and tumor boards. 3 about implementation of drug trials, and 4 about dissemination. 5 about transporter access and 6 about training. It's 2 years. It started 1st of January this year and will run for 2 years. These are the countries that are involved. So besides the Nordic countries, we're happy to have experienced countries on board like France, Portugal, Sweden, Germany, Croatia, Italy, Estonia, Hungary, Lithuania. And we see now that many countries are joining, and we are open to that. Of course, we cannot refunnel money, but you can all work and use the protocols and give your input. So Ireland, UK, Belgium, Greece, Romania, many other countries are getting on board. So yeah, this just to be short, because we're running almost out of time. There are 2 co-leaders of each work package, and we have equal gender balance. So we have a harder time finding men than women, because one work package leader man is on 2 work packages. But this is really about getting the things in order. Work package 2, molecular diagnostics, NGS-based test, clinical decision support systems, you have heard about that, and the MTBs. It's really critical to have the basis in order when you want to proceed with this. Then we have the implementation of DRUP-like trials. So it's important to build a data aggregation platform. We are now planning on having stage 3 studies together, but there are so many cohorts in the DRUP trial. My hope would be that we can even combine stage 1 and stage 2 cohorts. And then the implementation dissemination, well, but talked about that. So the added value of the advanced diagnostics test, the cost effectiveness of the treatments by itself, and the cost of implementation, etc. So that's really an important thing to do. Then we have the equitable access to these studies for patients. So Bettina from Melanoma Patients Network Europe is driving that together with Ulrich Lassen, and well, it's important to have this in place, and indeed we already did a first workshop on that. And then education. So Loic is taking care of Shanif's work there very well in Iwana, and I mean, this is really important. I mean, I see it in my own country. The level of knowledge is going up through molecular tumor boards. We do that through Teams at 8 o'clock in the morning, so every physician can call in while he is in his car driving to his work or whatever, and it's getting increasingly well attended. So there's more and more interest, and the physicians, oncologists in training, they all join as well. So I think it's good, and we hope we can spread that through Europe. So Primrose, that's the follow-up grant that will start the 1st of July for 5 years for 6 million. And you see already that many more countries are on board here. Not all are beneficiaries, but we work together and we'll find new grants to do this. And the countries also have to look within their own country for grants, of course. There's a lot of interest from the pharmaceutical companies for this concept, by the way. So yeah, so DRUP-like trials. Well, I explained that to you. It's really a platform trial, and this I explained to you as well. So the different stages. Stage 0 is also important. So you have to look at the available evidence from preclinical studies, from case reports, and it could be negative information which leads you to decide that the patient will not enter into stage 1. So the work before entering stage 1 is also very important. So this is what Primrose looks like. You have a work package on external control arm. It's quite difficult, but it was mandatory in the call to have an external control arm, so it will be a synthetic control arm from many databases that are available to build further on the data platform. And then there is money for the cohorts, especially for 4 stage 3 cohorts. So that's good for a kickstart for countries that need to start. Per patient, 1,200 euros, I think. Ethics is important. Defining novel patient-relevant endpoints. We do that together with patients, but also we are in contact with the EMA. And for instance, the magnitude of clinical benefit scale from ESMO, the guideline committees. So we have to work together to predefine good endpoints. Then the living lab is important, that you have just, that Ebba just has shown that slide. The health economics is important, and then again, communication, dissemination, and project management. So this slide on the left hand, there's only a couple minutes left, but Ebba showed a few. I just want to point out the other ones that she didn't tell yet. I think the health insurance companies can make a favorable impression by supporting this, and I think the MSI story in the Netherlands pays off for the insurance company who funded this risk-sharing part. Economics is important for the private partnerships. Pharma is in a very interesting way to get your drug reimbursed for ultra-rare indications. So there's a buy-in for pharma. Innovation agencies, they can be demonstrator, and policymakers, they can also have an opportunity to observe and learn from this model, and this could also be implemented outside of oncology. Clinicians are happy with treating their patients, and researchers can publish a lot about that in high impact papers, but the most important thing is they can also fine-tune what we give to our patients. So I think there's a lot of patient-centered benefits from this. So these are the data set we can use for this synthetic controls. The Hartwig data set of about 10,000 patients can be added to that because that's an open source. These are the studies in the countries where they run. In of course the Determine, and in Ireland the study, and in Estonia should be added to this list, which is growing. And this I will skip because this is really boring with the what we need to do. So in conclusion, precision oncology is one of the way forwards in oncology. I haven't talked about immunotherapy, but there is an end to what we can achieve, but it's certainly an addition. So don't expect too much, but don't do it because you think it won't work, because it works. Treat less, rapidly developing field. There's many hurdles, but it is possible through collaboration. And then this one again. So really for the people at home. Also, if you go there, then you can also attend the PCM4EU meeting that is adjacent at the end of the meeting. And you can also request online participation in the PCM4EU. So you can send an email if you wish. Thank you very much.